Thank you for joining us on Synthesis Workshop. My name is Duc Tran, a member of the Synthesis Workshop editorial team. In today's installment of our Advanced Chemistry course, we are joined by Duc Tran. Duc earned his PhD from ETH in 2014, studying rhodium-catalyzed CH transformations. He then pursued a postdoctoral research fellow position at the University of Cambridge, after which he joined Janssen, where he currently works now as a principal scientist. Today, as part of our Special Topics module, Duc will be providing an overview of process chemistry and share a few recent examples of successfully completed scale-ups. Thanks a lot for joining us, Duc.
Hello everyone, my name is Duc Tran. Today I would like to give you a short introduction about process chemistry and highlight a few examples of the type of work from process chemists around the world. By definition, process chemistry involves development and optimization of production processes for chemical compounds and the scaling up of laboratory reactions. It is usually associated with pharmaceutical industry, but there is also significant process development in agrochemistry, ingredient for perfumery or raw chemicals industry. One of the most common questions that process chemists receive is that the molecule was already prepared by discovery chemists. Why not just increasing the scale? Increasing the scale is indeed what we focus on, but it is not as simple as you might imagine, but needs a lot of ingenuity and broad understanding of different fields of chemistry. We sometimes need to redesign the synthesis, as the initial synthesis is not the most efficient way, because discovery chemists need to focus on speed and diversity of the molecule. Secondly, upscaling needs to take into account several additional factors such as safety, reactor design, mixing, heat transfer, waste stream treatment, and so on. Last but not least, we need to decrease the cost of goods of the desired drug molecule and ensure the sustainability of the production processes, which usually goes hand in hand. We usually follow the general principle of green chemistry, such as using catalyst, decreasing catalyst loading, use environmental friendly solvent, avoiding unnecessary workup or isolation, and so on. You can learn more about this field by following and reading our dedicated ACS journal named Organic Process Research and Development, where I took most of my example from. It is the standard timeline for drug development from discovery until the patent expiration. Process R&D should start when a few hundred grams or more of a fragment or API is needed. So usually start from the preclinical development, where we start to build a common core in a large amount to be able to enable the SAR study. Later on, when the API structure is defined, we are responsible to deliver API for clinical study of phase 1, 2, and 3, and later on design the synthesis for the commercial scale and prepare registration and documentation for the launch of the drug in the market. Being a process chemist is very rewarding and impactful. First of all, you design the scalable synthesis of the target or the API itself. You need to explore a broad chemical space in order to develop the optimal synthesis, of course, within the time frame of the project. Secondly, you need to deliver the API batches for clinical study. It means that the API that you prepare will be used in clinical study to cure patients. In that way, you feel a direct impact on patients. And lastly, by developing the commercial process and bring the drug into the market itself in order to cure a lot more patients, you feel you have a major impact on the drug development itself. Of course, you cannot do all this work alone. You need to collaborate with a broad team with different expertise from the safety, analyst, technology group, production team, engineering, crystallization unit, and a lot more. Teamwork and good communication is an important skill for the process chemist. From a practical point of view, process chemists work on a very broad spectrum, from milligram scale up to kg scale in a kilo lab. However, at a high level, we can classify the work into four different stages. The first stage is the route scouting, when the molecule just entered the team or the department. We will work and try to find, if possible, a better retrosynthesis, more suitable for upscaling. At this stage, we use a lot of high throughput experimentation to try to screen as broad as possible for the disconnection, try to find a proof of concept for the new route. Usually we work at milligram scale as the molecule is not largely available. The second stage is reaction optimization. Once we have a route defined, we start to optimize every step of this route. It's very similar to the work of the methodology in a PhD organic synthesis. We use a lot of focus and high throughput experimentation to optimize the reaction, try to find the best base, the best solvent, the best catalyst to achieve the highest assay yield and the cleanest impurity profile. We start to upscale to gram scale, also to assess on sterility and a few other factors. The third stage is the process development, where we start to upscaling the reaction even further, 10-100 gram scale, and try to define the workup and the isolation of the product. Here we need to define what kind of workup needed and what solvent composition is needed for the crystallization, or if telescoping is a possibility. At the end of this stage, usually we will have a process that is suitable for upscaling if needed. The fourth stage is process optimization, where we will try to fine-tuning all the parameters, try to make the process as efficient as possible. To approach it, we can do either one factor at a time or by design of experiment to optimize several parameters at the same time. This last stage is very plan dependent because we need to develop the process to fit for a certain type of plan. That's why it's very project dependent as well. At the first stage, the route cutting phase, once the team start to work on a certain molecule, we will start right away with the idea collection for different retrosynthesis of this structure by either discussion with colleague using retrosynthesis software or discussing with the MedChem team have already experience with this molecule. The most challenging work for this stage is the ranking of the round because it's very project dependent and depending on the timeline and request of each project. There are several parameters that can be used to rank a round, such as the internal knowledge. If the company have already some experience with this molecule or this some type of transformation, it will be very useful to leverage this internal knowledge. The next one is also very important is the cost of good projection because we want to have the cheapest synthesis route possible. And the synthesis need to be scalable, which is linked to the PMI prediction. So PMI is the process mass intensity, so it's the amount of chemical used to produce 1 kg of product. And as you can see in the reference, there's several PMI predictions have been published to highlight the importance of this prediction for ranking. The next one is, in some case, the most decisive parameter, which is the safety of the reagent and also of the process. In some case, we can use new technology such as flow or photochemistry to handle some hazardous reagent or some radical pathway. Once the ranking is finished, then we need to plan the approach. Should we try one round at a time or screening several reactions at the same time. That will depending on the size of the team and the time that we have for this project. For example, the BMS team need to synthesize brand protein. The team has several disconnection possible, and in the paper they describe different disconnection and the use of HD to quickly select the right path forward for each disconnection. And here I would like to highlight only the synthesis of the highly substituted indole core. As you can see, just to make the indole, there's several possible pathways. Either reductive strategy where you use butanedione with a metal and hydrogen, or a neutral strategy where you use hydroxybutanone with a Lewis or protic acid to perform the cyclization. And the last one, the oxidative strategy, where we use butanone, which is the cheapest, but you will need to have a certain type of catalyst and an oxidant to form the indole ring. At the end, based on the H3 screen and the internal knowledge from BMS, they have selected the neutral strategy where they use hydroxybutanone, and from the screening, the diphenyl phosphoric acid delivering the core indole with very high yield and high impurity profile. The second stage, reaction optimization, it is a rather common task for process chemists. Usually we use a lot of high throughput experimentation to optimize the reaction, the chemical component of the reaction, but we need to run gram scale reaction to start to observe some physical behavior, which usually is the root cause of the scale-up effect. These are calorimetry of the reaction, exothermic, endothermic, the homogeneity of the reaction, which is very important for the stirring in the and operational timing, how long it need to heat up the reaction, and so on. We need to understand these key parameters to be able to assess the robustness of the reaction. In the plant, every operation need to be in our range, so our reaction mixture need to be stable in that time scale as well. For some special cases, such as catalyzed reaction, we need to have a deeper investigation, such as kinetic analysis to confirm the mechanism, understand the reaction mechanism, identify unstable intermediate, identify some pathway, the catalyst activation or deactivation, to be able to prevent undesired pathway. As an example, I would like to highlight the work of the AstraZeneca process team, where they have described very detailed the upscaling of a CH correlation of the 2-chloro-6-methylpyridine in order to synthesize AZD4635. In this paper, they need to scale up CH correlation, which have been demonstrated in the Chem synthesis using substoichiometric amount of B2Pin2 because the side product, the pinacone borane, can also run a CH correlation. However, hydrogen is a byproduct which can cause some safety concern. So the team have started with a high throughput experimentation, and they can quickly identify catalyst loading can be reduced from 0.25 to 0.025 mole%, a tenfold reduction by properly form the right catalyst, which ensure the ligand chelation on the iridium. Secondly, they can use the in-process analytical tool such as Raman, NMR, and HPLC to follow the reaction kinetic, and thus they can conclude that the relative reaction rate of B2Pin2 is much higher than HPPin, and both reaction is oxygen sensitive. That gives a very important information for the process safety, because in that way they can use a stoichiometric amount of B2Pin2 to completely avoid the hydrogen evolution of the second step. And also by understanding the byproduct of the process, they can develop a quenching method for the waste layer to improve further the safety of the overall process. And this process have been upscaling to multi-kg to be able to afford the molecule for the clinical study. For example, the process chemists in BMS need to develop synthesis of the deuterated nitropyrazone. They prepare it from deuterated methanol, making it into a living group using torsion chloride and sodium hydroxide at room temperature, and then telescope to the next step, adding nitropyrazone using the sodium hydroxide existing as a base to achieve a very efficient methylation of the pyrazone. However, when I analyze the reaction mixture, they find very high level of impurity C5 deuterated as shown here. It can achieve up to 15 and 20%, so it's non-negligible. By carefully depicted the whole reaction scheme and all the byproduct, they find out that this deuteration is due to the formation of HOD, which is the byproduct from the first step. And this impurity is shows very difficult to purge during workup because, as you can imagine, have very similar solubility profile as the desired product. However, the process chemists have used exactly the way that this impurity is formed to destroy it. So as this C5 deuterium is formed during the reaction because of the strong base sodium hydroxide in presence of the HOD, which can scrambling that CH position. So during the workup, they use also the same base sodium hydroxide with a large amount of water to scramble back to the desired product. So instead of wasting 15 to 20% of impurity, we can convert that impurity into the product during the workup, so to improve the yield and the impurity profile at the end. And the last operation, and also the most critical in the process, is the isolation of the product. Of course, before developing this step, we need to ask ourselves, is the isolation really needed? And if not, is telescoping a possible option? Of course, telescoping will improve the efficiency of the process by reducing the PMI. However, it also means that we will bring all the impurity profile and byproduct to the next step. It's for sure something to check. And if the isolation is needed, depending on the state of the product, it can be either distillation or crystallization, and both of them is a field by its own based on their complexity. In pharma, most of the time it is the crystallization that we need to develop to isolate either the intermediate or the API itself. From my personal experience, it's only when I start to work in the process chemistry that I start to encounter the polymorphism. Polymorphism is behavior of an organic compound where it can be isolated under different form with the same purity. These different forms have different physical property, and these properties are very important for the isolation process and for the API, also the biological activity of it. For example, a new form of API can result in a new pattern for the drug. And just to give you a flavor of the complexity in the crystallization, I would like to show you my own example in the synthesis of odalasphere. The pinnacle ester shown here is an important intermediate before engaging in the Suzuki coupling that needs isolation. By doing different experiments in the lab, we naturally find out three different forms of this compound. Form 1, Form 2 are anhydrous form, and Form 3 is a monohydrate when we have some water present. As you can see in the same picture, Form 1 is plate-like, very good for filtration. Form 2 is nido-like. It can be filtered very poorly over several days in 100 kg scale. And the best form is Form 3, but it to be prepared in a very unique condition. And these three forms can be converted in between them. For example, form 2 is the thermodynamically most stable, and it is more stable than form 1. So if we stir form 1 long enough, it will eventually convert to form 2. Form 1 is kinetically preferred. So if we crystallize the product in an anhydrous condition, form 1 will be formed first. But then from the first statement, it will slowly convert to form 2. Form 3 is the most thermodynamically stable in presence of water and a certain temperature range. So to be able to develop the most robust process, we need to design the crystallization where form 3 remain the most stable form during the whole isolation process. So we need to design a solvent swap, but making sure the content of water is always in the right range. These were just a few selected examples of process development. If you would like to learn more about this field, please refer to the reference that mentioned on the screen. Of course, every OPRD article will be a very interesting case study for you to become familiar with process chemistry. I hope that you will be interested in this field and will become a process chemist one day.
Thank you for tuning in to this installment of our Advanced Organic Chemistry series, and thank you, Duc, for sharing this overview of process chemistry. If you enjoyed the episode, you can support us by subscribing and telling your peers about this podcast, and feel free to send us any questions or comments that you have. Follow us on Twitter to stay up to date, and see you all next time.